Welcome everyone to Cures and Capital, the podcast on value creation in biotech and pharma. I'm Martin.
And I'm Simon.
And today we are talking to a really special guest, Jack Scannell. He is the godfather of the Etheros law. He worked as a neuroscientist, a strategy consultant, an investment analyst, and he is now a CEO of Etheros Pharmaceuticals. He is really the jack of all trades, and he has deep insights into the world of biotech and pharma. Simon, what can our listeners look forward to in this episode?
Fasten your seatbelts. This is going to be a very densely packed discussion. First, we'll go over the past, present, and future of R&D productivity, and there's no better person than Jack to take us through that conversation. We will discuss return on investment on innovation in biotech and pharma and why that has been under pressure for virtually decades. Jack will explain to us why that might be so and what we can possibly do about it for the future. Then we will discuss decision quality, decision making, and the tools at our disposal for making better decisions in biopharma. We'll discuss two sets of models, disease models, animal models, and their validity and what it means to our decision-making process in biotech and pharma. Then we'll discuss financial valuation models and why they're not perfect and what we can potentially do about it. And we'll try to make it all practical so you can put it to use in your daily lives.
At the end of the episode, we will go over mine and Simon's reflections and takeaways. And we will try to unpack how we believe we can use these insights into our daily lives. Before we deep dive into the episode, we want to emphasize one thing, and that is that all the views and opinions expressed in this podcast are our own, and they do not represent those of our employers.
Without further ado, enjoy our conversation with Jack Scannell. So, Jack Scannell, welcome. It's a pleasure and an honor to have you on the podcast.
Well, thank you very much. It's a pleasure to be invited. So thank you.
It's maybe just to start, because I'm a little bit curious. I saw on your profile on X, maybe we need to help our listeners unpack that bio a little bit, but it says science, drugs, money, interested in making better R&D decisions.
Yeah, so, well, you're meant to write something kind of short and pithy, right? So people have a rough idea about what you're interested in. But I've been interested in biomedical matters for a long time. I trained in medicine a very long time ago. As I say, to the great benefit of the English NHS, I never practiced in medicine. I went and worked as an academic neuroscientist, where I did a mixture of visual neurophysiology, sort of proper wet science, and also sort of computational neurophysiology. And from there, I went to work for a company called the Boston Consulting Group, where I did a lot of fairly commercial stuff in drug companies. From there, it was effectively the European equivalent of Wall Street. So I worked for an investment firm where I looked at drug and biotech stocks. I was one of those people whose job it was to try and guess whether the stock prices were going to go up or down. I then went into biotech for a couple of years, doing biotech drug discovery with some technology that was closely related to that I worked on during my academic career. A couple of years really focused on R&D productivity before going back into investment, and then in 2019 going back to the R&D productivity world. And then from there in 2022, 2023, moving to spend most of my time with a small biotech startup. And I think the common interests have been, as I said, science, drugs, and money. And I think a particular thing that I got very interested in is decision quality and the tools that we have at our disposal to make decisions. R&D is a kind of serial decision process. And I did some work, which we may talk about later, that gave me the view that one's ability to do it well is exquisitely sensitive to the quality of the tools you have at your disposal to decide whether therapeutic candidate's going to be good or bad. That may sound obvious, but I think in quantitative terms, or rather the quantitative impact isn't obvious. So hence the pithy Twitter/X title.
Yeah, no, I think that really summarizes it very nicely. And right now you're the CEO of Etheros Pharmaceuticals, and I couldn't help notice that, and people can go to the website there, but the chemistry there is really interesting one, and even just for the chemistry, people should visit the websites. Maybe it's kind of soccer ball chemistry. Is that?
Yeah. I think when I talk about our company to people, there'll come a point where if they're a medicinal chemist, there'll be a sharp intake of breath, and maybe they'll fall off their chair. So we have some very, very unusual chemistry. But I think it's actually been backed up by some extremely rigorous science over about 25 or 30 years. Yeah, we have some compounds that are small molecules, but they mimic a number of enzymes involved in protecting tissue from inflammatory and oxidative damage, and they're orally bioavailable, brain penetrant.
Super cool. And so just to ground our listeners a little bit, back in 2012, you published this article diagnosing the decline in pharmaceutical R&D efficiency.
Well, so I think, I'll talk about the motivation first, and then I'll kind of tell you what it was about. I think the motivation was a sort of dissonance between relentless scientific optimism, right, on one hand, and the grim reality of the economics of the drug industry circa 2010 on the other, right? So I'd spent my, formative professional years as a biological scientist, trying to get grants from the government by telling them how brilliant basic science is. And if only we do a bit more basic science to understand biological mechanisms better, then boy, oh boy, we won't be able to control that huge flow of new treatments, right? I'd been through or exposed to the sort of genomics hype in the sort of late 90s, early thousands. I'd worked for consulting firms who wrote reports telling their drug industry clients how, if you follow our advice and implement the technologies that we say are hot, you'll have so many successful drugs you won't know what to do with them, right? So I'd sort of seen the optimism. But then when I started working in investment around 2005, it became very clear that the productivity trends were heading in the opposite direction. So the number of new drugs per $1 billion spent in 2010 was roughly 100 times lower than the number of new drugs per $1 billion spent in 1950, when we didn't have all of these wonderful technologies, right? And although declining productivity was a matter of debate in many areas, my view was that the debate as it was didn't really address the ugly contrast between declining overall productivity and the improving efficiency and quality of scientific inputs. And much of the debate was actually rather self-serving. So again, if you're a consulting firm who did organizational redesign, you would diagnose that the productivity problem was you hadn't hired us to do your organizational redesign. If you're an investment bank, you'd diagnose the problem as you haven't bought enough biotech companies lately, right? And my view is that many of these diagnoses seemed very, very unrelated to what the underlying problems probably were. So that was the sort of motivation behind them.
Just so everyone is on board with E-Room's law, because you're kind of one of the godfathers of that, and maybe you hate to talk about it, but let's get it out of the way.
Well, no. If people remember it, so I'll talk about it. So there's a thing. Gordon Moore coined the term. He was a pioneer of the computer semiconductor industry. A law was named after him, Moore's law, which related to the exponential improvement in efficiency of certain elements of computer hardware. I think it was the transistors per chip doubled every certain number of years. I've forgotten precisely how many years. And this has held for a very, very long period of time, over decades. So you have this exponential improvement in efficiency. And E-Room is more spelt backwards, and it was a sort of joke to describe the fact that the number of new drugs approved by the FDA per $1 billion spent roughly halved every 8 or 9 years between 1950 and 2010, right? So the computer industry was obeying Moore's law forwards. The drug industry was obeying it backwards, hence E-Room, which is more backwards.
Mm-hmm. So how do we measure R&D productivity?
Well, one short answer is not as well as we should, right? So I think the sad truth is the measures that probably society would be most interested in are very, very hard to make. I would say there's no consensus about how they should be made, and it's not even clear to me that they can reasonably be made, right? So really a society would be interested in, the number of quality adjusted life years per unit of R&D input, right? For a whole bunch of reasons, we don't really know that. So we're stuck with sort of proxies, and I think some of them are pretty simple and pretty concrete, right? So one measure that I used, because it was easy to derive, not necessarily because it was the best measure, was the number of new drugs approved by the FDA per $1 billion of R&D spending. Now, even that's a bit of a tricky measure because, you've got to define R&D spending, and is it global or is it in the US, et cetera, et cetera. But one could at least produce long, consistent time series of data, right? There are other financial measures, actually, which are more narrowly industrial. So they may not interest society as a whole, but probably interest shareholders, that actually one can derive pretty well on a backward-looking basis, right? So there's a whole bunch of measures that relate R&D input investment to, branded pharmaceutical industry profits, and you can do that at the company level or at the industry level, right? And I think those measures are fairly sort of straightforward and unambiguous. Another problem, though, with those measures, I think both the financial value and the social value of pharmaceutical innovation is very, very heavily skewed, right? So again, if we look at the number of drug approvals per $1 billion spent in recent years, well, if I'm honest, most of those drugs are drugs for fairly small populations, and many of them are not hugely effective, right? But then thrown into the mix, we've had, GLP-1s for obesity, we've had mRNA vaccines for COVID, right, which are right very useful for very wide numbers of people. So there are a lot of genuine measurement problems. But I think on the basis of most sensible measures, setting aside those kind of lumpiness problems, whether it's the narrow financial measures or number of new drugs per $1 billion spent, there was a big, big fall, from 1950 to 2000. And since 2000, or since 2010, it depends a bit which measures you look at. So the number of new drugs per $1 billion has actually stopped slowing down. That's bouncing around because the FDA started approving more drugs circa 2010. Probably to do with the molecular slicing and dicing of oncology and more rare disease drugs. And if you look at some of the financial measures, they look pretty good up until around 2000 because drug market growth offset the increase in R&D investment. But since 2000, the drug industry has kind of stopped over-earning, and its financial returns in R&D have sort of normalized to levels which, at least on some measures, don't look particularly attractive.
So no matter how we slice and dice, or if we use whatever metrics, it still looks pretty a sinister view here. So the next question is, so what is the reason that is a downward trending curve?
Well, the number of drugs per $1 billion spent down from 1950 to 2010, and then it's kind of flattened. And if you look at financial returns, flat from 1960 to around 2000, and then declined. So I think, there's a number of reasons, and we wrote about them in the 2012 paper. But I think there are two big ones I would highlight. And the first one, we termed it in the paper something called the better than the Beatles problem. So the idea here is the analogy of popular music. So, imagine how hard it would be to launch and commercialize new music. If every record had to be better than the Beatles, if you could download all the old Beatles records for free, and if you didn't get bored of listening to the Beatles, right? If you had that conjunction of items, it'd be quite hard to launch new music. But in fact, in pharmaceuticals, you do have a very, very similar problem to that, because today's blockbuster is tomorrow's generic. So it does become virtually free, and then doctors don't get bored of prescribing it. Right, so sort of metformin, right, would be a good example of a pharmaceutical equivalent of the Beatles. And what you find is that in the therapy areas where R&D has been historically successful, they get increasingly crowded with almost free generic medicines. And what that does is that effectively undermines the economic incentives for more R&D in those areas, and it pushes the R&D investment in the areas where historically the drug industry has been unsuccessful. And a bit later on, we might talk about why the drug industry has been unsuccessful in those areas, and I've got views about why it might have been. But for, whatever the particular reason, those are probably the areas that are likely to be hard in one way or another, right? Otherwise, the drug industry wouldn't have found them difficult for the last 100 years. So this is a huge quantitative effect, right? Because if you look in the US today, 90% of prescriptions are for generic medicines, right? So there's large swathes of medicine where actually we know we can discover quite effective drugs, but actually, because we've got so many quite effective drugs in those areas, there's no point doing it. There's no commercial point anyway doing any more R&D. And I think the very second obvious trend, and this isn't meant actually to be a criticism of the regulator, but I think it's just a statement of fact. The regulatory environment has transformed since 1950, right? As I've probably said in other interviews, lots of the things that were done in the 1950s, they wouldn't simply annoy the FDA if you did them today. They'd wind you up in jail if you did them today, right? I think there was a remarkably different attitude, a sort of tolerance towards effectively people being experimented on, and a whole bunch of other regulatory requirements which now exist and didn't. And that inevitably made R&D much cheaper. It reduced cycle times, and it allowed a whole bunch of discovery methods which today are very, very difficult. And I suspect those are the two big ones. There are some others as well, but if I had to name the big two, it would be those two. And I think the other thing I'd point out is, and again, this comes back to the 2012 paper, I wrote that partly because I thought the sort of public discourse around declining R&D productivity wasn't as good as it should have been, given the importance of the problem. So if you want good explanations of Eroom's law, they have to be powerful and progressive, right? They have to be able to explain an order of magnitude of productivity change. And I think, right, and they also have to explain why things have got harder over time. So lots of things that might very well help R&D productivity are not components of the explanation of Eroom's law. So I'm sure organizational design matters, right? But I don't imagine that organizational design was brilliant in 1950 and is terrible now, right? So I think I was particularly looking for things that were sort of progressive, cumulative, and could explain orders of magnitude of change in productivity. And if they couldn't, they might be important, but they weren't as important.
That makes sense. If you then kind of fast forward and you look over the last decade or so, there was a publication you made called Breaking Eroom's Law, made a status update. You co-authored an OECD report on R&D productivity as well. So where are we today?
So I think some of the measures look better, but some don't, right? So there was a clear inflection around 2010. So running up to 2010, roughly, drug industry costs were sort of growing every year. And the number of drugs approved every year was bouncing around at roughly 25, right, by the FDA. What's happened since then is drug industry costs have probably continued to grow, but the FDA started approving about 50 drugs a year. And that, I think, is to do with a change in the things the drug industry is focused on to a certain extent. So it reflects a different problem choice, right? So we've had molecular slicing and dicing in oncology. So, what used to be thought of as a single cancer type is now, recognized to be a bunch of different genetic subtypes, and drugs are targeted at those different genetic subtypes. And we've also had, I think, much more in the way of rare disease drug discovery as rare disease drug pricing has made it more attractive. So that's been a clear shift. What hasn't shifted, though, is the narrower measures of financial return applied to the drug industry. So what's typically happened is that the R&D costs have continued to rise, and although more drugs have been approved, they've been approved for narrower indications.
Yeah.
Right. And I haven't refreshed my numbers for 2024, right? So that's a kind of view up to 2023. I'd be surprised if anything radically different has happened in 2024.
Yeah.
I think that has been a great explanation. And I think, Jack, before we move actually into, I think now we go to today, what does this mean for the future? I'm actually wondering, when you released these analyses a decade ago, almost now, and more than that, how was it received? You mentioned the topic of public discourse. Now you told essentially the entire industry they are not doing well. How was it received? And has it in any way changed over the 10-year period?
I think I'd be lying if I pretended it had more influence than it's had. Right. So I'll give you two slightly non-serious examples before maybe talking about slightly more serious ones, right? So after I published the 2012 paper, there's a period when I got invited to go and talk to big drug companies, right, to their R&D organizations or their finance organizations. And it was about this kind of relentlessly depressing Eroom's law stuff. I'd go and talk to them. And initially I thought, wow, they must really like the analysis I've done. And then someone said, actually, Jack, they've only invited you here so it's that the R&D guys don't ask for a pay rise, right? So you always have to wonder, if you've done some analysis, what people are doing with it. And then similarly, I see lots and lots of biotech pitches, and a lot of them, a gratifyingly large number, have an Ehrum's law graph, right? At the start. And there's an old sort of there's a very good political satire on British TV called Yes Minister, where they talk about something called the politician's fallacy. And it runs as follows. You see a situation, you say, this is terrible, right? Or rather, no, the situation is terrible. We must do something, right? And then someone says, well, this is something, therefore we must do this, right? So I often see the Ehrum's law analysis followed by, this is terrible, we must do something. And then the something is whatever that company would have wanted to have done anyway, right? So now to slightly more serious answers. I think some drug companies realized, probably in parallel, right, that some of the things I was writing about were interesting. So for example, if I look at AstraZeneca's 5R program, which I don't pretend for one moment was influenced by anything at all I did, when I look at it, it strikes me as very sensible, right? But I suspect that's convergent evolution, right? And then I think there was a second strand of work which coincided with the Ehrum's law work, which I think had more of a niche audience, but didn't have quite the same meme value. So shortly following the Ehrum's law work, I got very interested, or actually also published as a supplement to the Ehrum's law work. I got rather interested in representing the R&D process as a sort of formal search process. You make a simple mathematical model of the R&D process. Okay, so there's lots of things we're looking for them. It's kind of search process. If we represent that quantitatively, what kind of parameters is search efficiency very, very sensitive to, right? And I wrote some things around the nature of search in chemical spaces, and also the nature of differentiating between good and bad therapeutic candidates. And I think that work was kind of less accessible and it didn't have the meme value. But occasionally I have had people, or rather, I think in some ways, in certain niches, that's probably been more influential. And occasionally, people tell me they've found it helpful, and that's very, very gratifying.
Okay, so you didn't meet with a lot of, no, Jack, you're wrong. This isn't a problem for us.
Apart from with a few details, surprisingly not. And I think to a certain extent, people kind of knew there was a problem. And I think what the 2012 Ehrum's law paper did, it was a kind of clear statement of problems that many, many people knew existed, but most of them couldn't write about because because if they wrote about it as part of their day job, they'd have got told off by someone. Right, I was an investment analyst, so my employers were quite happy for me to write about it. Lots of people in the drug industry, probably their employers would have been less happy for them to write about it, at least for external consumption.
A quick question, because and also to I mean the extent today to which this is at present, but the cognitive dissonance, because you see, okay, there's a declining trend, Ehrum's law, but on the other hand, you see in the media the companies are making a profit, all the R&D folks are getting paid. So should I really believe this, or is it just an artifact, this analysis?
Yeah, so it's very interesting. So my firm view is it's not an artifact, right? So, and I think there is an error that critics of the drug industry make, and arguably the advocates of the drug industry. I mean, it's quite a hard point to advocate, right? But, and again, this may be more accounting than people want to hear, but there are certain accounting artifacts that make the financial performance of the drug industry look much more attractive than it is, right? Or so to put it another way, the drug industry is actually quite capital intense, and capitalists, i.e. the people who own drug companies, are really interested in returns per unit capital per unit time, right? They're called capitalists, not profitalists. So profit margins actually. So if you look at the profit margins in the drug industry, R&D is expensed. It doesn't appear on the balance sheet. If I was building a cement factory rather than building a biotech, and I was spending that money on building a cement plant and not on R&D, it would appear on my balance sheet, right? So when I look at, so it would appear as kind of capital on my balance sheet. So if I'm looking at return on equity measures or capital efficiency measures, my capital efficiency looks much lower than a drug company that actually has exactly the same cash flows, and that's purely an accounting artifact. So if you treat R&D money like capex, which effectively is the economic role of R&D money when spent in the drug industry, the financial performance of the drug industry looks much worse than it does if you just expense it. And I think many sensible investors, when they're thinking about return on investment in the drug industry, they think about the R&D as capex. And it looks to me, although, so you've got this funny artifact where drug industries can appear very profitable, but at the same time, they're returning below the cost of capital, right? And again, it's a slightly complicated accounting point.
Super interesting.
But it also always meant when I was an investment analyst, when drug companies always told us how fantastic their return on equity was, I'd say, well, it is if you don't calculate it properly.
Right. But I think that's also a big problem in the industry, because to the broad public, it seems like the pharma is just making a huge amount of money.
Yeah.
Well, if we really account for how it should be treated, then all of a sudden you have an issue where everything you invest in, or most of what you invest in on average, is actually losing money, right? And there you have this disconnect.
I do think there's a communication problem, but I also think that there's a problem here that the political debate is very polarized. So actually, generally what happens is people pick their position and then they find the analysis that supports their position. And also, I think on this point, it's not really in the interest of drug industry management to tell you, actually, our return on equity is lower than you think it is. Right. So the drug industry has this difficult PR problem in that they have to convince governments that if drug prices were a little bit less, it would be a disaster, while at the same time convincing their shareholders they're doing really well. And it's quite hard to have those two, to simultaneously hold both those views.
Yeah. So Jake, I'm wondering if we now look ahead, right? So we talked about the past, we talked about today. I wonder, how do you see the future of R&D productivity, and especially for the next 10 years? Is there a bright future? Should we expect the decline to continue? Or is there something we can actually see already now that we are turning the trend a little bit?
So I think the Beth and the Beatles problem, that ain't going to go away, right? That is a sort of reality of the drug industry that's going to kind of persist forever. I also think that there should be a strong Bayesian prior against assuming that the next big technological thing is going to solve the problem, right? Because we've seen lots and lots of big technological things that would improve R&D productivity. There's a long list from 1950 to 2010, right, of things that everyone thought would be wonderful. And then it turns out actually the productivity trend was pretty sort of steady in the face of those. This may improve R&D productivity, but not in the way that necessarily biotech companies like, right? So, I think it does appear that the world has a place where R&D can be done more cheaply than in the US and Western Europe, and that is becoming increasingly important, right? I recently read that big pharma did roughly a third of their in-licensing deals with Chinese companies over the last year. Now, from a consumer surplus perspective, right, that may be a great thing, maybe not such a good thing for Western biotech companies. One other thing I would say is that and you're asking me to sort of prognosticate, right? I think prognostication's really hard, not least because it seems to me that the sort of big innovations come from where lots of people aren't looking, or have historically come from where lots of people aren't looking. So today I was just thinking about what have been, probably the three biggest drug classes of the last 25 years. They've been anti-TNFs, they've been the immuno-oncology drugs, PD-1s, PD-L1s, and they've been the GLP-1 analogs, right? I don't think any of those were kind of central to mainstream thought, right? So the anti-TNFs were effectively failed septic shock drugs repurposed by Mark Feldman into rheumatoid arthritis, which was a field which at the time many people in the drug industry thought had minimal commercial value, right? The PD-1s were kind of largely invented, or some of them were invented for doing other things to do with immunomodulation And effectively they were sort of repurposed when it became clear, I think, from the CTLA-4s that there was something in immuno-oncology, which until that point had looked, like it really wasn't going anywhere. There'd been sort of decades of immuno-oncological failure. And then the GLP-1s, they didn't come out of the white heat of sort of genomics, right? They came from sort of classical gut hormone physiology, right? So I think I'm constantly struck by the fact that the positive surprises seem to come from where people aren't expecting. And also returns are incredibly skewed, right? So a disproportionate amount of the value of the drug industry is created from a very, very small number of assets, right? So I'm going to step away from prognostication, if you don't mind.
Entirely fair. Maybe one comment also, if you look at your analysis, Jack, and the history, at least the history will say that high throughput screening, combinatorial chemistry, genomics, that didn't make a dent in R&D productivity in the industry. So the expectation should be, all things being equal, that AI will also not make a dent.
Yeah, so actually, I think genomics has made a dent actually in some productivity measures, but it's made a different dent to the dent that was originally conceived. So I think what genomics has done is it has allowed us to slice and dice and characterize certain pathologies in such a way that we can match the models to the pathologies, but the pathologies have been smaller, or rather rarer, than would have been expected, at least by the major proponents of genomics in the 1990s, right? So it's kind of, it's not, I don't want to be, that's a great benefit for patients with those particular pathologies, but it's not what the original promise was, right? I think with AI, again, I don't pretend to be, again, I'm not going to pretend to be an expert here, right? I've sort of done enough to make a fool of myself talking about things I don't know about, right? But I don't know enough to know about this properly. But if I had to, but I think where I'm optimistic actually is around slicing and dicing and characterization of human pathology and measurements of human pathology and characterization of human disease, more than sort of bottom up scientific push. Right. So, and again, AI is a term that is applied very broadly, right? And I think in its broad application, it's not a very helpful term. But I'll give you, and it may be some people wouldn't regard these things as AI, right? But for example, I was talking to a neurologist the other day who was involved in measuring endpoints in a clinical trial in a neurodegenerative pathology. And they were using a conventional endpoint, which was, I recall, a 6-minute walk test, right? And they were looking at the effects of pharmacological treatment on that. And they said to me, the main driver of the 6-minute walk test for this patient population is what time of day they have the appointment. Because if they get an appointment first thing in the morning, they can park right next to the clinic, and it only takes them 30 seconds to walk to the clinic. And that means in their 6 minutes, they can walk quite a long way. Whereas if they have an appointment in the afternoon, they're right at the far end of the car park, and that's 400 meters across the car park. And by the time they get to the clinic, they can't really walk at all, right? And that's the nature of the endpoint that is being used in a sensible, serious trial. And I think there's a whole bunch of pathologies, particularly sort of neurodegenerative, psychiatric pathologies, where actually that's what endpoints look like, right? Well, if you have sort of modern ways of measuring movement or patient behaviors or continuous monitoring, suddenly actually you've got a whole bunch of ways of measuring things that are going to be much more sensitive and specific, and will make clinical trials possible, and will give you positive results in a way that things like a test that basically depends on how far the patient parked from the clinic, it won't.
Let's transition our conversation into decision quality, decision making, the themes you have been preaching. And the word preaching is notice you have used the word preaching. Is around the predictive validity of disease models. So what is that?
So I'll give you a very little bit of background first. So I alluded to maybe 10 minutes ago that I'd done some work over the years on kind of trying to model the R&D process in a slightly more formal way. And then you can understand the parameters to which R&D productivity is sensitive. And one of the one way of modeling this is you say, well, okay, imagine that we're trying to find ligands which could be drugs against a particular target, or we're trying to find targets which could be good targets for a particular pathology, right? Well, let's take the ligand example. okay, we've got a very large number of compounds which we either have made or could make, and we've got a bunch of ways of deciding whether or not those compounds are going to be useful or not in people. And really what we're trying to do is we're trying to sort of, so you could imagine representing these therapeutic candidates in a sort of measurement space where, one dimension is, your high throughput screening potency. Another dimension might be, your sort of computational, toxicity results. Another dimension might be, how well it does in a rat model of the disease, right? And of course, not every candidate gets tested in all these models, but at least notionally, you could think about representing your therapeutic candidates in this measurements, this sort of high dimensional measurement space as a sort of points in this high dimensional space. And if you create that formalism, you can start looking at the trade-off between different parameters, particularly the parameter which the measures correlate with each other, and then more importantly, the degree to which they correlate with the human outcome of interest, which you could think of as the sort of the score against the primary endpoint in your phase 3 trial, right? As a sort of, that might be what the truth state is, right? And if you model the R&D process in that way, what you find is that your ability to detect good candidates is very surprisingly sensitive to the degree to which the assays, or I call them decision tools, output would correlate with the human outcome of interest in the unlikely event that you had infinite money and no ethics. I.e., if you could have put all these candidates through your rat or through your high throughput screen and then through the person, the thing that really matters is the correlation coefficient between the model system and the human outcome of interest. And we call that predictive validity. And there's a number of ways of operationalizing it. And for a lot of the analysis I've done, I've operationalized it as the correlation coefficient that would exist if you did that infinite money, no ethics experiment. Right.
Yeah. And what numbers are we talking here in terms of the quantitative relationship? Is like a 99% or is it like 1%, 50%?
Well, so let's suppose, you're searching, or what you find is that for sort of searches that are relevant to the R&D process, where maybe 1 in 1,000, 1 in 10,000, 1 in 100,000 candidates might be useful, changes in the correlation coefficient between your assay and the human outcome of interest of 0.1 can have the same productivity impact as doing 10 times or 100 times more brute force work. Right. So, and I used to be a sort of, working biologist, and I would plot graphs from my experiments, and sometimes the correlation coefficient would be 0.5, sometimes it would be 0.6. And I'd say, well, actually, there's no real difference between those, right? Well, actually, if I was using those models for drug discovery, it could be one of them would be 100 times more efficient at detecting good drugs than the other one, right? And the issue here is that correlation coefficient is generally is almost always unmeasured, and it's generally unmeasurable, right?
Yeah.
But it's important. So anything that sort of decorrelates the relationship between your model system and the human outcome of interest is likely to make your performance much worse, even if it massively increases your throughput. And there's some nice historical examples here, right? So, for example, very low throughput screens of antimicrobials done in the 1930s in live mice seem to find some useful antimicrobials, right? Massively high throughput screens done in the 1995 2005, where people tested 10 to the 7 compounds in, across, 100 high throughput screens, didn't find anything. And actually, if it turns out the correlation between the results in a septic mouse and a septic human are 0.8, and the correlations between binding affinity and an isolated protein in your high throughput screen and a septic human are 0.2, you would expect the best compound out of 200 randomly selected and tested in the mice to be better in people than the best compound out of 10 to the 7 randomly selected, tested in the low correlation assay. You'd expect the 1 out of 200 tested in the mice that look best to be better than the 1 out of 10 to the 7 tested in the assay that look best, right? And I think, so coming back to the R&D productivity headwind, right, and Eroom's law, one hypothesis I have, which I think is, well supported by the historical data, is that in operational terms, the diseases for which we have treatments are the diseases for which the models have tended to work, right? In the 1950s, that was because the models were people, right? So the first 2 classes of antidepressants were effectively tested by phenotypic screening in patients, but actually patients are not a bad model of depressed patients. Very small numbers of compounds were tested, and we found some that worked, right? But over time, and some of the models were animal models, right? So spontaneously hypertensive rats, at least I've been told, are not a bad model of hypertension, right? Some rodent models of diabetes are probably not bad models for diabetes drugs. But the therapy areas for which the models are generally predictive are the therapy areas where we've got lots of drugs. So those become, they are then rendered commercially redundant. And we're left with the therapy areas where typically the models don't give us good drugs, right?
Yeah.
So Alzheimer's, solid tumors, et cetera, and ironically, because those models don't give us good drugs, we keep using them.
Yeah.
Right. So there's been a sort of selective enrichment for bad models.
So, and the models we have that are good, they have now been exhausted, if I understand you correctly. And it's not easy to get new models, right? It's a massive effort for academia, industry, validation. It's many years. So what do we do? Is it experimental medicine? How do you see us moving forward here?
So I think unsurprisingly, it's a mixture of things, right? So I think the first step is recognizing the quantitative importance of model validity. So no one working in the drug industry thinks a bad model is better than a good model, right? But unless you run the decision theoretic maths, you don't realize quite how much better a good model is than a bad model. And I think I wrote a paper in 2022, which I think tried to talk about this in more practical terms. It was based on actually having some very good collaborators who tried practically to do this, many of them in the drug industry, and also doing some sort of practical consulting work with drug and biotech firms to sort of think about practically how do you think about it. And I mean, yes, so step one is understanding the quantitative importance. Step two is probably just having some lingua franca, right? So again, one problem you face is if you talk to if you talk to a pain biologist, an anti-infectives person, and a neuroscientist, right? They'll all have completely different ideas about whether models are any good or not, right?
Yeah.
So and they won't even have common terminology to talk about models. So, for example, in psychopharmacology, there's a bunch of concepts around sort of model quality. People talk about face validity, construct validity, predictive validity is a term that would have been used in the psychopharmacology literature.
Yeah.
I wouldn't say I've hunted all of the cancer model literature, because I haven't, but I don't think those are terms that are typically used, for example, in the oncology model literature, right? So just the way people think, so it's quantitatively important, you need common language, and then there's a whole bunch of practical frameworks that you can use to evaluate models. So this is not a counsel of despair. It's not like we actually have to test millions of drugs in rats and then all in people. There's a whole bunch of things you can do. So, does your model have appropriate statistical and experimental hygiene? Are your sample sizes big enough? Have you got risk of bias? There's then things like, are the endpoints relevant for the endpoints you can measure in people? To what extent are you recapitulating the relevant biology? There's a kind of sort of clever checklist approach, right, which one can use to try and nudge model choice in the right direction, cognizant of the fact that actually even small improvements in model quality can actually can be a big deal.
I'm actually wondering, putting on like a practical head, if I'm a executive in a pharma company or in a biotech company, now I know about this problem, right? So now I'm possibly using models that might not be giving me what I actually want to have out of them. Does it mean, or would you then suggest that I should now actually say, okay, I'm pausing now. I'm not going to use the models. I'm going to start investing in better models before I actually continue with my research? Should that be what the executives should be focusing on before they start investing into using bad models?
My view is straightforward, is in some cases, yes. Now, the industry is incredibly heterogeneous, right? So I've met lots of people in the drug industry who know more about their models than anyone else on the planet, right? But it's interesting, I've met people in the drug industry like that, whose knowledge about their models has not then found its way into decisions, right? So you've got lots of folk knowledge or craft knowledge about what you believe and what you don't and why. But actually, I think many, I'm sure not all, but some companies don't have the mechanisms to reflect that in the investment decisions that get made. So I'll give you one of my favorite examples here. And I don't think this isn't always very difficult. There is a drug called Terilazad, which was positive in a large number of animal stroke models. right, for ischemic stroke. And if you think about it, ischemic stroke, although there are actually major differences between the structure of rodent brains and human brains, if you think about it, ischemic stroke, you'd imagine if you cut off the blood supply to the part of brain of a rodent, the subsequent pathophysiology is not going to be that different to the pathophysiology if the blood supply gets cut off in a person, right? So there was a bunch of positive Trilizad studies, and then there were some spectacularly failed human trials of Trilizad. And there's some very nice sort of meta-analysis work done by a group in Edinburgh under a guy called MacLeod. And what they published was that if you look across the animal studies of Trilizad, the median delay between inducing the stroke in the animal and administering this drug was, if I recall correctly, 10 minutes. If you look at the human trials, the median delay was, if I recall correctly, and I may not recall precisely correctly, was 5 hours. Right? So in this case, if I go back to how you think about models, right, you want to make sure that you recapitulate the biology, you want appropriate statistical and experimental hygiene, and you want the tests and endpoints to match. Well, here's a case where actually the tests and endpoints are completely different, right? So why would you expect an effect after 10 minutes in an animal model to predict an effect after 5 hours in a human? Now, and look, a lot of people in the drug industry do this brilliantly, right? But clearly some people don't. And I think there's a whole bunch of reasons why it's not done well right often. I think there's kind of availability bias. You typically do the model the way it's always been done or the way the CRO does it, right? It's quite hard to do something different and new. I mean, that's one obvious thing. I think another thing is that people haven't run the decision theoretic maths, right? You don't realize quite how important it is to try and do it properly. So I think a huge amount of expertise exists. But I think that expertise doesn't always get captured and reflected in decision processes, right? No. And because model quality is quite hard to measure, it's often under-evaluated and doesn't weigh sufficiently in decisions.
Also, I think you captured really nicely. So step one is the model literacy, how we talk about models in our organization. Next step is to try to integrate it in the decision making. If you could offer maybe one very sort of practice-oriented piece of advice. So let's say we're going into a decision meeting whether we should invest putting our drug candidate in the clinic. What sort of conversation can we actually have?
So I think, again, it's probably easier for me to illustrate my thinking with concrete examples, right? So, and I'm not always close enough to detail to know whether my concrete examples are good, but they illustrate my line of thinking, right? So if I think about depression, right, this has historically been a very difficult, it's been in recent years, it's been a very difficult place to deploy R&D capital. Because the animal models don't seem to be terribly predictive of what happens in people. And actually, unlike some other therapy areas, the small and early human trials don't seem to be very predictive of the later human trials, right? So one thing that's really interested me, and I don't say it's interested me enough that I'm an expert on it, but it's interested me substantially, are sort of effectively behavioral biomarkers for certain sorts of psychiatric illness, right? So patients, for example, who are depressed have a bunch of symptoms, but one thing they do is they rate the positiveness of facial expressions differently from people who aren't depressed. And that is something that seems to normalize very quickly if you give them antidepressant treatment, despite the fact that standard depression rating scales probably won't register a change for about a month. And also standard depression rating scales have very high placebo response rates. Right? So in this case, you think, well, okay, could I run a study that was structured in such a way that I pre-screen compounds using this behavioral biomarker, right? So I would throw away any compounds that don't have an effect in the behavioral biomarker. And then before I run my trial, I will pre-test patients on the behavioral biomarker, and only those who have a changed facial response would I then randomize to drug or placebo, right? So what you do there is you use an experimental medicine step to enrich the candidate set of compounds. And then what you do is you use another step to effectively to recruit the right kind of patients, right? And you improve the signal to noise ratio of the process, and maybe you don't have the same problem with high placebo response rates, et cetera, et cetera. Now, I don't know enough about depression to know really whether, the extent to which, that is being done or is practical. I know people who work in depression who do that sort of stuff, right? But I'm not an expert in that field. But I think another way of thinking about it is to think work back from the human pathology, right? So I've always thought, and again, this is a sort of illustration rather than necessarily a reality, but imagine you want to do neurodegeneration. Maybe a sensible thing to look at would be chemotherapy-induced peripheral neuropathy before you do Alzheimer's disease, right? Because one can probably satisfy oneself that chemotherapy-induced peripheral neuropathy in a rodent has the same pathophysiological basis as chemotherapy-induced peripheral neuropathy in a human, right? And one could and you could test that, right? Because you can get tissue from humans who've had surgery. You can get, and then you can then work back from that. So, okay, what would be a good model of the chemotherapy-induced peripheral neuropathy in the whole rodent, right? There may be some way I can simplify it and make it a bit more tractable, but still satisfy myself, right? So, and then another third way I've sort of seen it done is really just with kind of clever checklists. So, for example, I've done some work with some Alzheimer's experts who effectively, you go through an exercise to characterize, almost you do a target model profile. Okay, so for Alzheimer's, what would the characteristics of a effective model have, right? What pathophysiological features of the human disease would it recapitulate? What would it be its statistical properties, signal to noise ratio, repeat, if I repeat it, how similar does it have to be? Can it measure the endpoints that I'm interested in, right? Whether they're real endpoints or sort of surrogate endpoints you might measure in a human. And then what they did is they scored the available models against that checklist.
Mm-hmm.
And what they found was that the available models then clustered into several groups.
Yeah.
Right? Do we know if any of those models are particularly good or better or worse than the others? No. But what you could say is if I want to advance something in Alzheimer's, I want to try it in each of the in each of the clusters, right? Rather than just one.
I think it makes sense. Yeah.
So I think there's a number of practical things that can be done, but it's much easier to answer the question with specific examples.
No, definitely. I know some companies also work with a target research profile in contrast to the target product profile, and that's maybe a way to sort of make this operational.
I think, and you start with the target profile before you select the models, right?
Yeah.
So you prospectively say what would good look like, and then it's easier to decide whether the model's any good or whether you think the model's any good or not.
Can I ask, Jack? Because now we are talking, and I wouldn't forgive myself if I didn't ask. I'm a finance person. We are talking about decision-making tools. We are talking about models. What is your view on valuation models, the holy grail of finance? Do you think it helps our decision making?
So I think sort of DCF-based pipeline valuation, right, has a number of, I think, critics, right? Nonetheless, it's the industry and arguably the venture capital standard, at least when you reach a certain stage of the development process. I have a specific criticism of NPV-based models, and I think that an analogy here is the AstraZeneca 5R framework. There's a couple of very good papers written by folks from Astra, and they talk about truth-seeking versus progression-seeking behavior. And current DCF models, you would argue, are kind of progression-seeking models. There's a very important parameter in the model, which is called probability of technical success, which is the assumed probability that I go from, let's say, preclinical to phase 1, or phase 1 from phase 2, or phase 2 from phase 3. And you use the probability of technical success parameters to kind of work out the cumulative probability of each step, which tells you how much money you would expect to spend in each step, and then also the cumulative probability that you get something out at the end, right? And you sort of weight your cash flows by those cumulative probabilities. Now, I think actually there are much better ways of doing it, and I've sort of published some of this, but only in supplements in obscure places, and hopefully I'll publish it a bit more thoroughly sometime soon. But if you think about it, probability of technical success is a dumb measure because it conflates good and bad decisions. So if I say the probability that we go from phase preclinical to phase 1 is 30%, that is actually a mixture of good decisions to progress good candidates with bad decisions to progress bad candidates.
Yeah.
And if I suddenly improve the quality of that decision step,
Yeah.
the mix of the portfolio in phase one is going to change. But if I simply use probability of technical success in my model, there's no way of reflecting that change, that value, which will flow through the rest of the portfolio, and it will change the numbers of candidates and the quality of candidates in all downstream steps. It will change the number of phase 3 trials you do. It will change the number of candidates to get approved, right?
Yeah.
So my view is you need to throw away probability of technical success and you need to replace it with a parameter with different parameters, which are the assumed true positive rate and false positive rate of your decision step, and your assumed probability that the candidates at that decision step are any good, right? And there are certain ways of parameterizing that. Again, which are buried in a supplement of a paper I wrote with some folks from Emulate. But I do think if we parameterized our DCF models like that, we would start encouraging people to do the right thing, because you could show your venture capital fund or your portfolio manager that by improving the decision quality of this step, I'm actually improving the value of the product, or I'm improving the value of the project.
That's very interesting. So, Jack, you're also now a very hands-on practitioner of R&D productivity and so on. You have analyzed the industry, and now you are a CEO of a biotech company, Etheros Pharmaceuticals. So how has that changed your view now that you're actually sitting in this position and you make decisions, model validity, return on investment?
Yeah, well, so I did a bit of drug discovery around sort of 2012, 2013, and I learned very hard then that drug discovery isn't as easy as investment analysts think it is, right? It's genuinely difficult. And I think we're a small company, right? So actually, I think this is going to be the first thing I've learned, I think, is a sort of, I would say, small company perspective, right? Well, actually, there may be more general. I think actually problem choice is really important, right? So I think a lot of academic work is around, okay, I want money to study disease X, right, or disease Y. I'm going to do the science of disease X or disease Y. Then I'm going to spin out a drug for disease X or disease Y. I spend most of my time thinking, given what we think the attributes of our compounds are, which problems should we be studying? i.e., where could one learn things that would increase the value of the assets in a way that is practical and efficient, right? So what shouldn't I, where is it that, for example, I could spend a huge amount of money and a huge amount of time and I still wouldn't have, and I still wouldn't have got any closer to knowing whether this is likely to work or not, right? So it's thinking about that sort of the informational, it's the sort of conjunction of information and capital and cost and time, right? Where are we going to play? Where aren't we going to play? I think that's, I would say, the sort of thorniest thing I'm grappling with, and it depends on a bunch of things. It depends, for example, on the validity of preclinical models. I would rather play in those areas where I think the preclinical models will tell us something useful. But actually, if the preclinical models aren't going to tell us anything useful, I'd rather play in areas where actually you'd learn from relatively quick, fast, inexpensive human studies, provided the tox looked okay and it was sensible to do them, right? And I think that's the sort of the thing that I'm really grappling with. And my sense is probably, I don't know whether you call it a luxury, but I think a lot of people in the drug industry probably don't have the luxury of that, right? So if you're working in a company that's already got a big franchise in area X, you probably don't have the luxury of problem choice, right? But I think I think that for me is the thing that I'm really spending my time thinking about.
No, and I think this is very actionable. Also, for every company putting together an R&D strategy, pick your disease areas wisely. Think about the validity of the models.
Then the early human trials. Exactly. Yeah.
So if you were to give one piece of advice for R&D leaders out there, if they want to improve R&D productivity in the organization, what is it that they should take to heart?
So I could say something grand, but then it would also almost certainly be non-actionable and probably wrong. So I'm going to be very narrow, but it's something. It's a real issue actually. I, in my last days as an investment analyst, was absolutely amazed by quite how crowded some therapy areas are. Right? Look, doing models well is difficult. Right? Biology is unpredictable, but it's actually not that hard to count how many other programs there are in a therapy area. Right? That's pretty easy. Right?
Yeah.
And I was chatting to someone today, an oncology R&D expert I know called John Hickman, and he was looking at RAS, which has been a very fashionable target in oncology. And he told me that on his latest count, there were 52 RAS programs either in preclinical or clinical development, right? So my one piece of advice would be, don't be the 53rd RAS program.
That's extremely actionable. So thanks for that advice, Jack. I know you have a similar question, Martin.
Yeah, and in light with the focus of this podcast, right, we want to make sure we educate both sides. So now I'm just going to ask, and you have very much financial background as well. What would be your advice to the other side, to the business and finance folks in the company?
Oh, you mean to the finance people in drug companies?
Yes.
Yeah, I think my advice actually to the finance people would be look out for ways of doing things better than thinking about probability of technical success in the models. I think explicitly try and break out the quality of decision steps, assume true positive rates, false positive rates, and the probability that assets are likely to work. And if you do that, you can build financial valuation models that actually reflect the actionable things that people in R&D try and do. So if you think about it, if you're doing R&D, right, you're either trying to pick good chemistry or good therapeutic candidates a priori, and then what you're trying to do is you're trying to accurately decide which ones work and which ones don't, right? And it would be really good if financial modeling aligned with that, not just some aggregate that mixes up and mashes together good and bad decisions and then is blind to their downstream consequences.
And can I just ask, actually, now I thought it's all clear, but now I actually have a follow-up question, because I fully understand it, but I also kind of struggle. So when we have a drug, and let's say the drug has multiple potential target profiles, right? And now I'm actually not sure how to use your insight. Should I then not use the valuation model to basically say, this area actually seems to have a better payoff? I will rather focus my resources and efforts here. Should I try to not use the financial models for that?
No, so I think one uses financial models, but one structures them differently. I think there's several different things that we may be talking about. So one is the difficulty of reflecting optionality in financial models, right, in the drug industry. And I don't know how well it's done, and I've tried to do it a bit. And in general, and I haven't tried to do it a lot, but I've tried to do it a bit. And when I've tried to do it a bit, you run into the problem that the model gets so complicated you can't explain it to anyone. Right. And I've read a lot of academic work on kind of thinking of sort of optionality in financial models. You probably know much better than me whether people are really doing that in drug companies, and certainly I haven't met many people who do it, right? Then I think there's a separate question, which is if one is doing DCF models, it's fine to do a DCF model, but the probability of technical success should be a function of your assumed true positive rate, your assumed false positive rate, and your assumptions about the quality of the candidate. It shouldn't be an input. It's an output, not an input. Right.
That's I have to say, I'm a finance person in the pharma industry, and this is very new to me.
And that was published as an Excel appendix to a paper I wrote with Emulate on liver chips. So there is an example in the public domain.
Okay,
but that's interesting. Yeah, we need to watch both animal models and valuation models. I think that's one takeaway here.
That's a great insight. But we are now coming towards the end, and now it's a chance for you, Jack. Is there something we didn't cover that you would like to say?
The short answer is no. I found this very interesting. I hope you have, but I don't. Yes. I don't think I hadn't had a chance to express myself. So thank you very much.
No, thank you also very much. And I have a question now. So what are you going to actually work on from now on? So maybe in 25, what is going to be your area of focus?
The big thing really is Etheros, the biotech. The small biotech CEO, I think, finds they spend a lot of their time capital raising. So I think 2025 is going to be busy knocking on venture capital doors. That's how I'm going to spend a disproportionate amount of my time this year, I think.
Yeah, we can't help asking because we love books. So what are you reading?
I read a lot of papers, but I
Mm-hmm.
read rather few books. And the one book I will recommend, it's a very peculiar book. So if you feel that life is going well and you're sort of content and happy and everything's fine, and you want to be a bit more miserable, then it's a book by a guy called Dino Buzzati called The Tartar Steppe, which is a book that made me deeply uneasy, but it's wonderful.
Okay, we'll check that out. So thank you very much. We'll of course link to all the your work we have discussed today. How can people get in touch with you, Jack?
Well, so the Etheros website, etherosPharma.com, or I'm also on Twitter and LinkedIn are easy ways to get to me.
Okay, thank you very much, Jack.
Thank you.
Thanks very much. Bye.
So this was an excellent conversation we just had with Jack. What do you think, Simon?
No, I completely agree. Very insightful.
Is there something specific you took away from it?
I think what I take away is that if I take on and consider myself an R&D person in pharma, so I admit R&D does not have perfect disease models, but I'd also then swing it back to you, Martin, and the folks in the finance part of this industry. You do not have perfect models either. Your DCF-based valuation models are not perfect. So I think it's a 1-1 on the score here in terms of model validity during drug development and valuation, actually. So I think that's one key takeaway from this.
And I think I'm going to annoy everyone in the finance world by admitting, I have to admit myself, the finance models aren't perfect. And I think one thing that Jack really pointed out is that the first thing he mentioned about the finance models wasn't all the things that finance people necessarily all think about. It's like, what is the discount rate I should use? What is this? What is that? The first thing he mentioned was, it's the way you treat the probability of success. And I think it also points out it's not necessarily how you make your model better by having better finance knowledge. I think it's how you make your financial model better by being educated more on how you should treat the other side. And I think that's a very powerful perspective to show up to the finance people and say, there's so much more to learn about the other side. And that's the way you can actually maybe make better decisions going forward.
Yeah, and I think, so what can we actually do? So it's not that you just pull excellent disease models off the shelf tomorrow and then we are better off. But what Jack also recommended is that start talking about your models and their validity in your organization that you can do. Maybe make a checklist for how you also bring that conversation into decisions around your assets. And then I think also to the point around probability of technical success, I think he made it very clear that probability of technical success, whether your molecule will succeed in the next phase, that is a composite of two very important dimensions. One is the sort of intrinsic technical quality of the molecule. Once you have put together that molecule, it will stay locked until it goes off patent. But the other part, the decision quality part, that's actually within your sphere of influence. You can practice that for every decision. And there's a saying that drug development is like 10,000 decisions in sequence. So, and that you can actually apply every single time, discuss what is the animal model basis for whatever decisions we're going to make here, or what is the data we have at hand here. I think that's very actionable, actually.
And I have to say, I have to admit on my side, that has been an area that I'm now a newbie to. So I think there's a lot for me to learn and really understand how to apply that in the real world. But I think one thing that became very apparent about this topic is that there are certain things in the pharma and biotech industry that can really have a profound effect, even if you just do a small change. And I think Jack really made it apparent that exactly this topic, the predictive validity of disease models, even just a small thing can really make a big dent.
Yes.
And I think that's a very important discussion to have. Is there actually something there we could do to capitalize on this effect and really make a difference?
Yeah. I think, and that's hard to operationalize probably, but he made a very, very clear point around the quantitative importance of model quality. So again, as you say, Martin, a small improvement in model validity translates into a huge impact in your R&D efficiency, that you could not get the same effect using brute force, for instance, by hiring more chemists or screening more molecules. That this model validity, that's where you can really move the needle. And Jack, he talks about not where you can get 1% increment in your R&D productivity or return on investment. He's very clearly talking about where can we see an order of magnitude potential for increasing productivity. I think that's, of course, you can improve your meeting culture, and that would give you maybe that 1 or 2%.
And I think we also have to have in mind that, and Jack excellently introduced it, is the R&D or the pharma and biotech industry has been on decline in terms of productivity and return on investment for a long time. Every 9 years, the productivity roughly halves. And there has been a long time that this has been going on. So now to really reverse it back, we really need to do big changes. It's not about small marginal improvements. It's about really moving the things that really move the needle.
Yeah.
And I think he made it very clear that maybe there are also alternative ways other than just doing AI that people should maybe focus on.
Yeah, I agree. And I even also mentioned that maybe the whole ecosystem is changing. Biotech in the US or Western Europe versus what is happening with biotech in China. So that was also something I actually pointed out. And then we talked the big themes. We briefly discussed AI. And also there, I think history tells us, and Eroom's law tells us that at least in the past, these new technologies did not give an order of magnitude improvement. So should we believe that AI is different this time? We have to see. But at least history tells us we shouldn't maybe expect too much of it. But of course, the jury is out, and I'm sure we will dedicate future episodes to discuss AI in drug R&D for sure. So that's one thing. And also now that we are talking about technologies, he gave some examples actually in the past where sort of classical pharmacology work, like he gave the example of the GLP-1 space, which is gonna be the biggest drug class the world has ever seen. And that came out of sort of solid scientific work and got hormone physiology, and then, but not super fancy technologies that completely caused the paradigm shift in the industry. And he says it was not mainstream thought. So that was an example there he gave. So that also shows there's still this serendipity, unpredictability in the whole process, that is not maybe that one technology that will then suddenly change everything.
Yeah, and I think in today's world, it almost seems like there's a lot of bets on AI being this one technology. And I think people are generally aware that yes, the productivity is declining, yes, the return on investment are being worse. And I see more and more that people start to rely on AI coming and save us. And I think we really just need to be, maybe it will not, but I think we really need to be aware that there's more than one way of solving this, and different levers have different impacts. And maybe this will not be the one, so we shouldn't just put all our eggs into one basket and hope that it will come and save us, especially as you say, if the history shows us that there has been other technological breakthroughs before, and we are still not getting better. So why should this be the one thing that really turns the needle?
Yeah, no, I agree. And it's also, maybe we don't need the perfect models. So Jack is very much, a champion for decision quality. So maybe also we encourage maybe people out there, try to experiment with whatever quantitative models you have. Try to bring them into your decision-making process, try them out and see if they work. Can you actually develop the models? Can you explain them? Do they enrich your discussions around your assets or not? And of course, we'd love to hear from anyone out there who's cracked the nut here and has maybe better disease models and better financial valuation models, or are better able to factor in optionality in their models, or how they talk about optionality versus classical DCF-based models. I also take away that it seems like we don't have the answer after this episode. What's really the path forward to do this excellently, besides paying attention to your decision-making processes, your animal and disease models, and not do your evaluation modeling blindly.
Yeah, I think even though we don't have an answer, I think there's this saying that path towards progress is by starting to admit that we are not there yet, or nothing is perfect yet, right? So you need to admit there's room to improve. And I think this is one of the ways, at least for me as a finance person, but at least also a person in the biotech and pharma industry, is that the models we have today are not perfect. So we really need to do something about it, or we at least need to be aware of the downsides of the models we currently have. We can't just think they are perfect, so we are going to force use them to make all the decisions we have. We really need to think hard about, well, knowing all these things about the models we are using, how can I at least try to mitigate some of the downsides and really use them in a way that they can inform decision?
Okay, Martin, so what are you gonna look into specifically tomorrow to understand better after this conversation?
And I mean, as we said, there's so much to really synthesize after. But I think the first thing I'm going to do, just because I'm a finance person, I'm going to follow up on Jack's paper about how to improve the financial models and really see what is there that I can take away. And then I definitely want to see what are the conversations out there. So what are all the different opinions about this topic? And maybe there is someone that figured out a way how to implement this practically. And I really want to see is there something I can do, maybe not tomorrow, but is there maybe something I can do the day after practically? How about you, Simon?
Jack already sent a bunch of papers that we will make sure to link to. So it was almost like the school teacher giving us homework. So there's a lot to dive into there around. Also, we briefly went over how also accounting of R&D expenses, CapEx, and those sort of things. And then I hope that some of our listeners will actually also join the conversation and reach out to us or drop a comment on the LinkedIn page. And as Jack said, drug discovery is genuinely difficult. So let's also admit that drug R&D, it is difficult.
Yes, I fully agree. And I just want to add one thing to what you just said, Simon. I think knowing you, we both like to read a lot. And I think sometimes it can stay really theoretical. Yeah. I think this is a perfect opportunity to really see what is out there. And maybe some people crack the nut. So I think we should really continue this conversation also outside of the podcast and see what other people think. So I really urge everyone to please come back to us. If you maybe read a different paper than we did and you found a different way of doing things, please do reach out because we want to learn. And sometimes it's very boring to just read the theoretical papers and not knowing how to apply it tomorrow at work. So I'm really looking forward to see if there's maybe someone else out there that is like me and Simon, that also wants to have a conversation.
And I think we'll not get around that. We have to go deeper. So, Jack, he said that Etheros law is quite of meme worthy. It sticks. A lot of people know it. At least now they know it after I've listened to this episode. Then you need to go one level deeper, and that's more towards the quantitative modeling, where the R&D parameters meet the financial parameters. So I think that's something we have to discuss more, Martin. But I think I don't have anything else to add. So please drop your comments on the LinkedIn page, and then we'll be back very soon.